Okay, I think we'd like to go ahead and get started. Mary, can you hear me okay with the microphone? Okay, we'd like to go ahead and get started. Hello, everyone. I hope y'all survived the storms yesterday and last night and that you have power. If not, I can sympathize. I was without power this morning and still don't have power at my house, but really want to welcome you to the Great Lakes Seminar Series, which is sponsored by the NOAA Great Lakes Environmental Research Lab and co-organized with them and the Cooperative Institute for Great Lakes Research. My name is Greg Dick. I'm the director of the Cooperative Institute for Great Lakes Research, and we're really excited to relaunch this seminar series. We had some time off from it as we had limited staffing, but we're delighted to relaunch it especially with the help of our new staff member, Margaret Throckmorton, who's been doing a great job of organizing this with Mary Ogdal's guidance. So we're also delighted that I believe this is the first in-person seminar since before the pandemic. So welcome back to the in-person seminar series. Hope everybody's in the room here is enjoying snacks. And I also want to extend a welcome to those that are joining us online from around the consortium, Sigler Consortium, and elsewhere. So today I am delighted to introduce our speaker, Andrew Ault. So Andy received his BA from Carleton College, and he first got interested in oceanography there at Carleton when he did a sea semester, which many of us have done, and took a sailboat between Hawaii and Alaska, and along the way measured dissolved oxygen and chlorophyll. So that got him going in oceanography. He then did his PhD in chemistry at the University of California, San Diego, where he studied individual particles emitted and transported in the atmosphere with real-time single particle mass spectrometry. And that was under the direction of Professor Kim Prather, who's at UCSD and also at the Scripps Institution of Oceanography, where Andy did parts of his research and coursework. He then moved on to do a postdoc at the University of Iowa, where he worked with the Center for Aerosol Impacts on Chemistry of the Environment, which is that's an NSF Center for Chemical Innovation that focuses on aerosol emission from marine source blooms and chemical reactions. And then he joined the University of Michigan as a professor in 2013. And he has studied aerosol emissions, in part aerosol emissions from the Great Lakes and inland lakes and their reactions in the atmosphere. And recently, we're delighted to pull him into cyanobacterial harmful algal blooms and studying the potential emissions of toxins into the atmosphere. So his group has identified significant concentrations of toxins in aerosols, different toxins, and peptides. That's what he'll talk about here today with this great title, Flying Slime, Great Lakes Aerosols and Harmful Algal Blooms. So Andy, thanks for joining us, and let's all welcome Andy here to GLERL.
Thanks, Greg. Everybody hear me all right? Excellent. All right. So thank you so much for the opportunity to be here. I've really enjoyed getting to chat with everybody over the course of Tuesday and Thursday. Also, I really appreciate the flexibility with respect to the timing. So yeah, I'm really excited to tell you about how aerosols and oceans and Great Lakes kind of go together. So as Greg was mentioning, sort of talking about where I've come from over the years, I've always been a chemist by training, but I've always kind of been interested in, what's going on with lakes and bodies of water and oceans. And so what we're going to talk about today are just these little bits of liquid that can get splashed up or emitted from different bodies of water, particularly inland lakes and the Great Lakes. And so over the course of the talk, maybe I need to click on here. There we go. I'm going to tell you about sort of aerosols coming out as bubbles burst. And so we're going to talk a little bit kind of actually about the physics of how a bubble bursts at the beginning and how that is different for a freshwater environment versus a saltwater environment. We're then going to talk about how we generate these aerosols in a representative manner in the laboratory. We're going to then talk about how we can actually go into the field and measure these. This is something, of course, of great interest to GLERL is getting out into the real world, and we try to do a bit of that as well. We'll then talk about how once they're up in the air, they will change as they're getting transported in the atmosphere. They can undergo reactions, they can go long distances. And then lastly, we'll tie it back into the harmful algal bloom work that Greg mentioned at the end there, where we're looking at can toxins, other peptides get aerosolized from harmful algal blooms. But all of this comes back to the idea that when a bubble bursts, you end up getting these little droplets. And I love some of these are actually from the Physics of Fluids journal. You can kind of see all these little bubbles right here, essentially as that cap disintegrates and as it falls apart, these are going to get launched up into the air. And if you go over the oceans, or 70% covered by oceans, and it's actually the largest source of aerosols to the atmosphere is bubbles bursting in oceanic environments. And so there's a lot of work that's gone into understanding this sort of in the marine context, but there's been much, much less in a freshwater context. And so we're really interested in, if this is, conductivity on this axis. I was talking earlier, I think with Russ about this. There was, we talk about concentrations here. I've just put it in millimolar. You can see, sodium and magnesium and potassium, all of our favorite ions in very high concentrations. This is a log scale on the left axis here. And if we compare that to what we have in the Great Lakes, we've done this by a letter of each of the different components here. So here you go, Herein, Superior, all these are from cruises done over the Great Lakes. And you can see that we have a lot less of this, these inorganic ions, and that the ratio is actually pretty proportional to dissolved organic carbon. So we have a lot more similar ratio. And that's going to lead to different behavior when it comes to things getting spit out of the oceans or the Great Lakes. And so it's actually kind of the way that folks have measured this over the years is really interesting that they'll actually use sound. And so as a bubble bursts, you end up generating essentially an acoustic signal that you can measure. And so people have used this to relate this back to essentially how big are the bubbles that you're looking at. And so if we look at the bubble radius here on the x-axis versus the number concentration for seawater, you'll see that we're somewhat shifted to the lower sizes here. And this is log scale on the bottom once again. And so we have really, really small bubbles that come out of seawater. But freshwater, you'll notice, ends up over here. You can see actually that we have this little hump here where things are bigger than a millimeter. So in freshwater, we actually get a lot of pretty big bubbles, which ends up making a big difference in terms of the aerosol that ends up getting generated. So we have big bubbles, but we have less salts. And so we kind of look at the balance between these two things. And so one of the things that we did when I moved here is I've been around oceans for a lot of my career. I'd never really worked on freshwater. Came to Michigan, and it seemed like a pretty logical thing to start trying to do. And so we have a little tank here. This is our our lake spray aerosol generator. And we can make, this is about 5 liters of water, 8 maybe or so, 4 or 5. And you can actually see we did some photography here to look at the sizes of the bubbles that we're generating. And so we can go grab water of any sort, inland lake, Great Lake, et cetera, put it in here, and we can generate aerosol bubbles or bubbles from aerosols that are the right size. And so this actually goes back to just a fundamental property of how bubbles behave in water. And so you can see here, this is a really simple experiment, but I always think it was kind of elegant, so I like to show it. Where in a low salt environment, essentially if you send 2 bubbles up through a capillary, those 2 will actually merge really, really easily. But if you have a high ionic strength solution, i.e. salt water with a lot of salt in it, the bubbles don't come together very easily at all. And so it's this difference in behavior that actually changes how much of the aerosol is going to come out. And so here you can see a picture, and this is, we actually had an undergrad who was really good at photography. We got a nice high-res camera, and so you can see these pictures Alexa got here of these saltwater solutions. And if we compare it to freshwater, just by eye, you can kind of see that these bubbles are generally quite a bit bigger than they were for the saltwater. And so if we count, and we automated it, don't worry, although there was some cross-checking by hand, it was, we had a great undergrad on that. You can see the concentration of bubbles per volume, essentially for synthetic freshwater, which is essentially, we took water, milky water, and added in ions to represent what's happening on the Great Lakes. This is real Lake Michigan water, and this is synthetic seawater. You can just see that we make a whole lot less bubbles for freshwater. And then we go to the more complicated figure. Here I now have bubble radius on this axis, so this is how big are the bubbles. And on this axis, I have concentration. And so if you look at the green, which is this either synthetic seawater or seawater from some of that data out at Scripps that I was involved with as a postdoc, you'll see that we have a lot of, the most bubbles we have are the smallest sizes for seawater. But in freshwater, it actually behaves pretty differently. And so our measurements are shown here as solid diamonds. You can see here some other measurements made. This was done by doing a tipping trough, where you essentially took fresh water, dumped it in, and then listened to the acoustics to figure out the size distribution. And you can see that you have a lot more of the larger particles for either synthetic or Lake Michigan freshwater. So we have a lot of big bubbles. But how does that connect to what's going on in the aerosol? And so here we can sort of do a cross check. Ah, okay. That'll work. All right. I'm always worried about being a little too loud, so we'll see how this goes. All right. So if we look at aerosols getting generated over oceans, this here now is aerosol size distribution. So on the bottom axis, we have diameter, and on the side axis, we have the number of particles per little milliliter of air. And you can see that for seawater, we actually have things right around 110 nanometers, and this is matches what people have known for decades, but no one had ever really looked at it for freshwater. And we actually see that as opposed to just having kind of one lumpy log normal mode here, we actually have both a larger mode at about somewhere between 180 and 300 nanometers, as well as a really small mode that we have there around 50 to 80 or so nanometers. And with that, we end up essentially having a little bit less aerosol getting produced, but we have a wider range of sizes. And if we compare back, the very first measurements, at least that I'm aware of aerosols over the Great Lakes, were made when Paul Shepson, who used to be at Purdue and is now the dean at SOMAS at SUNY Stony Brook, he flew over the Great Lakes and actually saw some modes that they didn't really understand, including one right here that they knew was coming from the lake, and actually this other one they didn't realize was coming from the lake, which correspond to these two modes that we see in our data from the generator. So we were pretty excited that this showed that we could identify where some of these particles were coming from. If they're in this yellow box here, that's called the ultrafine mode. Those are very, very small, and they don't have the longest atmospheric lifetimes. They often either grow or collide with things. But this accumulation mode, this one that's larger than 100 nanometer, once these particles are up in the air, they can stay there for days, even weeks. And so they have a long time to be able to do stuff or get transported, undergo reactions, etc. So the other way that we can make aerosols is with something called a marine aerosol reference tank. This one's a little bigger. This one is about holds about 40 or so liters of water. And it actually uses a plunging waterfall. And so the waterfall kind of like goes into the water and it generates the bubbles that are the right size. You might say, like, you're really obsessed with bubbles. Why do you care so much about this? But if you actually just put a frit down there and sort of send really tiny bubbles through, you end up not making aerosols that are the right size. You end up making stuff that would all be like over here and not particularly representative. So that's why we care so much about these bubbles. And so once these come out, we can collect them onto either filters or substrates and bring them back to the lab to look at them, or if we're in the lab generating it, we can look at them there. Okay, so we have the bubbles bursting. We're able to make these aerosols in the lab, but does this actually happen in the real world? It's all great to study things in the lab, but if it doesn't have relevance, then you're not really on the right track. And so, and I will say there's at least 2 or 3 slides. We have a Gurl wave height map. So thank you, Gurl, for awesome data products. Those are appreciated. And so this was actually kind of a spur of the moment thing. A grad student and 2 grad students and a postdoc went over when we had a big wave breaking day over on the west coast of Michigan, over here at Van Buren State Park. And you can see on this day, there were some pretty decent waves going on the Great Lakes. The nice thing about talking to this audience is normally if I'm talking to a bunch of people either on the coast or chemists, they don't realize that we get waves in the Great Lakes. It takes me about like 5 slides to convince them, no, like they really happen. But this audience knows that, which is fantastic and appreciated. And so here you can see the waves. And then what we're doing in this panel over here is looking at where the air was coming from at the site. This is using a different NOAA product, the NOAA high split model that you can use to look at where air masses have come from. And you can see the air was coming down the lake, which is of course why the waves are at the bottom. And we're going over where there was some pretty decent wave heights. And so the question was, well, are we going to see these aerosols that we saw in the lab that we hypothesized were happening? And thankfully we did, otherwise this would be a really short talk. And what you can see is we have a lot of calcium, calcareous lakes, not shocking, as well as some other ions, as well as peaks that are indicative of carbonate, although we don't directly identify that with elemental analysis. We just see carbon and oxygen. But we see these particles and they're right where we would expect them. And we even sent them into, Greg mentioned in the intro that I did single particle mass spectrometry as a grad student. And we collaborate with Carrie Pratt, who is also in the chemistry department at Michigan, who has a single particle mass spectrometer. And she was heavily involved with the early years of this work. And so we actually ran some water that we had generated this aerosol from into her instrument, and we were able to see some really nice mass spectral peak signatures for calcium carbonate as well. So we see the aerosols in the lab, we see them in the field, but now we can get to the interesting part. What happens when these things are up in the atmosphere? How do they change? How can they get transported, evolve, etc.? And so the first thing we did is we actually were making some measurements. Again, this is with Carrie Pratt up at the Michigan's Biological Station. And again, nice to be speaking to an audience that's probably all familiar with this. So we're right up here at the top of the mitten. And what we ended up doing were a couple of things, one of which is just trying to see what types of particles were there. And so Carrie's student, Matt, had published a whole paper actually talking about the ubiquitous influence of wildfire emissions, very topical this year with all the smoke we've been getting down here, on secondary organic aerosol, which is another really important atmospheric component. But when they put started getting the data together, we had this blue portion here, which was very clearly not smoke and not sort of haze from urban areas. And if you look at the sizes between 1 and 2 micrometers in size, which is pretty common for sort of these larger accumulation mode to coarse mode aerosol, you would see a lot of blue. And so that blue wasn't coming from sources that we were really expecting, which was surprising. If we look as a function of size, sort of in this 1 to 2 micrometer in size range, you can see that that's a solid, anywhere from 10 to 12% of the particles were coming from something we were not expecting. I'm sorry, that's just for the calcium rich one. We also had a sodium rich category that actually was, 20 to 30% of the particles. And so the question was, well, where did these come from and do they connect to the lakes? And so the first thing we did in my lab, a lot of what we like to do is we like to go particle by particle and look to see what they're made of. And so here we're using energy dispersive X-ray spectroscopy, or EDX, and we can see the different peaks. So in blue, we have calcium and then the 2 peaks for carbonate. So that checked out with what we had seen before. And then we saw sea spray aerosol actually, which were sodium. And the way you really know that it's sea spray is the sodium to magnesium ratio is 10 to 1, just like in seawater. And so at a single particle level, it doesn't matter if anything else has changed. If you have that ratio, it's almost always going to be from the oceans of some sort. And you can see it's not that aged actually, because you have a bit of chloride still here. If it's been up for a while, you'll lose the chloride. You'll just have nitrate and sulfate left. A little bit of sulfate here, and I don't have a label, but a little bit of nitrogen as well for nitrate. So we then also looked at the single particle mass spectrometer. So this is microscopy. We're shooting electrons at things, seeing what elements are there. Here we're hitting it with a laser and blowing it up to see what mass fragments we end up making. And we actually have some really nice peaks here for the calcium, as well as we see some peaks here for nitrate, which I'll talk about what's going on with that in a moment. We also saw sodium here, indicative of sea spray aerosol, but we didn't actually see a whole lot of chloride. We saw a lot of nitrate there as well. And don't get thrown by the fact that shows as nitrite. That's just a mass spec way that things fall apart with the laser. And so we saw that at the larger sizes, we had a lot of the sea spray, and then the lake spray was kind of distributed across a bunch of sizes. And so this all came out in the S&T letters in 2018. What we ended up doing is looking at where the air was coming from when we saw these types of particles and how much of a contribution did they make. So here we can actually you'll see I have this labeled as Hudson Bay, which was kind of surprising to us. But we actually did some modeling, this thing called positive source contribution function, which uses essentially that NOAA high split model. And we combine that with our time series of when we had our type of particle present. And we actually were able to show that the sea spray that we were seeing was coming from Hudson Bay, which was both surprising and pretty cool. Like I never thought of that as a source of particles here in Michigan before. And the other part that was nice is that if you look, this is the height of the back trajectory. And essentially what we saw was that it actually dipped down at about 48 hours back, which is right when the air mass was over Hudson Bay. So everything all kind of clicked together. And so this was, a decent portion of what we were seeing up there at certain times at UMBS. But the other thing we've been seeing was this calcium type that we thought was coming from the lakes. And so here we again, thank you, Goral, for wave height data. We have wave height data here versus where the air masses were coming from, whether it was over Superior in Michigan, just Superior, or just Michigan. And we were able to see that once again, we had a decent contribution over our size range here of about 0.5 microns to 2 microns of aerosol coming out of the lakes. And so we were able to see that these were transported. So if we went all the way back to here, this is about 30 kilometers or so. And so we saw that it wasn't just like a stray particle or two that it made. It was actually a decent fraction of what we were seeing at UMBS was coming out of the lake or even all the way from Hudson Bay. So that was pretty interesting. And the other interesting part of this is I mentioned that we see these peaks that are indicative of nitrate. So if you don't think a lot about atmospheric chemistry, we actually know from the dust chemistry world that calcite reacts pretty well with nitric acid. And so the same thing is happening, we believe, in these particles where calcium carbonate reacts with nitric acid, gives us calcium nitrate left in the particle, and then sends out water and CO2, which is pretty fun. And so we're able to see this here. And then we teamed up with Alison Steiner, who is an atmospheric modeler up at in CLASP on main campus in engineering, and her postdoc, Anna Farahani, and my student, grad student, Nicole. And what we started looking at was, could we actually see in the model, if we added in these particles that were coming out of the lakes, could we actually soak up a bunch of nitric acid coming from, say, Chicago or Milwaukee, and have that act as a sponge to go into all of these particles? And in fact, in that key middle size range, here her bin was specifically 625 to 2,500 nanometers. The red here is telling you that a whole lot of nitric acid is getting sucked up into these particles, which is actually a sink for NOx that people haven't really thought about before this paper came out in GRL 2 years ago. The other thing it does is actually ends up having somewhat of an impact on the pH because it ends up acidifying things. But because the calcium is in there, it doesn't partition out of the particles in the way that, say, ammonia or ammonium would as ammonia. And so and actually less ammonia and ammonium ended up in the particle phase as ammonium based on this. So that was both a surprising result, but also showed that if you're ignoring the lakes, you're not going to get air quality modeling correct in the sort of Great Lakes region. So that was pretty fun. And here, just to kind of give an idea, this is the extra amount of nitric acid lost in blue and the extra amount of ammonia left in the gas phase in red. So pretty exciting result for us. So the last part, the last part before the HABs that I wanted to mention is, so we've talked about how the particles get up, they can do these reactions. The other thing that they have the potential to do is to be really good at influencing clouds. So once again, we have our measurements right here. These are Goral wave height measurements or mapping. The wave height is here, buoy 45022, thank you. And you can see our back split coming from a high split back trajectory coming from over where the waves weren't. And so here we once again teamed up with Paul Shepson, who'd originally flown over about a decade earlier to find these particles for the first time. And he, along with Rachel Kerpus, a student in Carrie Pratt's group, worked with Nate May and Nicole Olson, Nicole in my group, to see if we saw these particles up in the clouds. And so essentially what they did is they sampled above the clouds as well as in the clouds. We sampled both cloud water as well as the particles, the interstitial particles in the cloud that weren't actually part of cloud droplets. And you can see here some of the meteorology, potential temperature, and the flight track of actually flying over the lakes. So we've kind of gone down this road a little bit because there had been some work done, published in 2008 from earlier than that, where they had done some flights over the Great Lakes looking at clouds and had been surprised by both some sodium chloride particles as well as some calcium particles that they had found in the clouds. They were, there's not supposed to be any oceans around here with salt, or sodium chloride. The calcium wasn't supposed to be up there. They actually suspected originally that it was mineral dust, which it turns out it was soluble, so it wasn't. And so you can actually see these. And so these are actual particles from inside of a cloud showing this calcium signature that we've been talking about. We also were able to generate aerosol from water collected underneath where the plane had been flying, and we ended up getting the same signature. As well as here's, we resuspended some of the cloud water, and we also saw that same calcium signature. So we kind of verified it 3 different ways. And so we actually end up then looking as a function of size, and we saw that once again, particularly at these smaller sizes, a lot of the particles were coming out of the lakes. That's the blue that you're looking at here as a function of size. And if we compared what was in the cloud water versus the ambient particles, there was a really great alignment in terms of this lake spray aerosol playing a role, or being a portion of the cause, I should say. Okay, so we've talked about how the aerosols get made. We've talked about the fact we see them and we see how they change things or get incorporated into things. The last part, and the part I realized that probably most of the folks here are most interested in, is how does this connect to harmful algal blooms and toxin aerosolization? So I don't have to, again, give like 5 slides of background about why we should care about HABs to this audience, which is very much appreciated. I'm sure you've all seen every single one of these images up here many, many times, but we care about harmful algal blooms. That's the key here. We actually, one of the first things we did when we started thinking about this is that Carrie student Nate went and collected water at 3 different sites, one over here in Lake Michigan where there wasn't a bloom going on, and then a couple here in the western basin of Lake Erie. And we brought them back to the lab. We used our little generators, and we just wanted to see, was there any difference in the aerosol that was coming out, depending on how much organic material was in the water to begin with. And we were pretty excited, actually. It was back when they sort of selected different articles for cover art, and we got to use them for that, which was pretty fun. And so we ended up sending, don't worry about trying to memorize all these labels. That's not the goal. The goal is just to point out that when we take the water, put it in the aerosol generator and make the aerosol, we end up having kind of 3 different categories. This will come up a couple more times. One is sort of the salt ones without much organic material. One is the mixture of salt with organics. And then ones where you have very clear markers for biological activity. And so when we look at these, we can kind of see as a function of aerosol size here, again, 1 micron, just to give a context, as well as the fraction of different things. We have a lot of the biological material in the larger sizes. We have a lot of the organic material that's probably, biomaterial that's degraded in the smaller sizes, as well as salts kind of across most of that size range. And so we ended up looking at this. This is particularly from right by the Maumee River input into Lake Erie, looking at these particles. And so if we compare, this one right here had the lowest, just a normal blue-green algae sensor. And then as we went to more and more higher concentrations of blue-green algae, we from the phycocyanate measurement, we were able to see more and more biological material in the aerosol that we were generating. And so that was pretty interesting. We also did some fluorescence microscopy, and you can see here the particles as well as how much fluorescence is present. So this is low organic biological material, higher and higher. We see more and more fluorescence. So here's blue-green algae versus fluorescence intensity for those 3 sites. And so the last couple of slides were just to convince you that when we get water from different, fresh water with different amounts of biological material in it, then we'll see a difference in what gets up into the air. So the next question became, well, what about toxins? Can we get toxins to get aerosolized if we take water from a harmful algal bloom? And so we went and we collected water. We actually were working with Eagle on this. Oh, they weren't called Eagle at the time, but working with Aaron Parker there. And he had collected water from both Mona Lake, where they were having a really big bloom, as well as Muskegon Lake, which was kind of relatively our control. And you can see here, this is both phycocyanate concentration, as well as we have both ELISA, as well as LCMS concentrations for the microcystins. And so that's with our close collaborator, Judy Westrick. She has an LCMS method that's really nice for looking at microcystins, not just total concentrations, but also a bunch of the congeners, which I'll talk about in a little. And so you can actually see over here on the right, we've got the structure of microcystin LR, as well as these are some of the different congeners that she was quantifying for us. You can see the different peaks from the liquid chromatogram, as well as the mass spectral fingerprints, just to kind of give an idea of how we go about doing this. And the key part was we wanted to see, do we get any of the toxins out of these two samples up into the aerosol phase? And so the idea with this is actually, we didn't just want to see if the toxins got up, but we actually wanted to see, did all the same toxins that were in the water end up in the aerosol equally? And to give away the punchline, they didn't. And part of what was going on is that if you have sort of the microcystin structure that we have right here, depending on which groups are at these two positions, here we have an arginine and a leucine, but depending on what you have there, you're going to change how hydrophobic or hydrophilic the particular congener is. And so if it's more hydrophilic, and you have an air bubble coming up through the water column, as it comes up, if you have something that doesn't want to be in water that's hydrophobic, it's going to find the edge of that bubble and it's going to glom onto it, right? So when it gloms onto the bubble, it's going to get enriched at that interface, such that when it bursts, you should get more up into the aerosol phase. And so here's what we saw in the actual freshwater for this particular monolake sample. We had a lot of RR, specifically some LR as well, other congeners also, but those two were dominant. And then when we looked at the aerosol, we ended up seeing that a lot of LR had gotten out, but almost no RR had gotten out. So we actually saw a shift in what people would be breathing in next to the lake versus if they were to drink the water. Obviously not that any of us would recommend that, but you get the idea, right? So this opens up a whole other angle that we need to be able to understand in terms of these harmful algal blooms. And so motivated by this result, Nicole here, who was the first author on this paper in ESMT a couple years ago, we went down in 2019 to Grand Lake St. Mary's, which is down in Ohio. I'll have a map on the next slide. And just some fun pictures of all the greenness. How many folks have been to Grand Lake St. Mary's before, other than Mary? Maybe one. Okay, so it's a great lake if you want to do this. It's only about 7 or so feet deep in a lot of places. It's very shallow, man-made, year-round harmful algal bloom. And so this is Nicole, and Isabel was an undergrad in the lab that summer. Ja, who's done a lot of the work you'll hear about, is right there. He's actually defending in 2 weeks, which is kind of coming up very quickly here. And so this is from his current manuscript that's under review at ESMT. And you can see this is the lake. That is the satellite image. It is always that green. And we measured the Wright State University, which is based out of Dayton, has a lake campus, which is right on the water right there. So that's where B and C are. One of those is right on their dock, and then one of those is on a porch at a little townhouse that we were all staying at. Here's a little island in the middle of the lake. We call this Prairie Creek down here, where they're actually using a lot of aeration systems to try to break the bloom up, which, if you've listened to the rest of the talk, you realize, oh, bubbles through air, through stuff might be generating aerosol, right? As well as the spillway, which you can kind of see here, and I have a better picture in a slide or two. And that spillway actually ends up generating a lot of aerosol, unsurprisingly as well. Oh, and here's Ohio. So, about a third of the way down, hour and a half or so south of Toledo. Okay, so the first thing we wanted to do is check the water. We have our ubiquitous green bottle water of lake junk, as well as our microcystin structure. Once again, Y and X just representing where we can change out these different groups to essentially change the hydrophobicity. And so specifically, there's also on some of the ones that we saw that were most abundant down there, we actually had, instead of 2 hydrogens up here, we'd had a methyl group pop on for some of these, as well as there's a hydrogen versus a methyl group in this position as well at times. And so there's other subtle modifications that can occur to these structures. And so when we looked around the lake, one thing you might take away looking at this is, place to place, the ratio of these congeners to one another is pretty consistent, right? We had a lot of this RR with a couple of slight modifications on it, as well as so the LR in blue here was just at the position, as well as our MC with the homotyrosine right here. So pretty consistent across the different locations. And so the first thing we wanted to say is, okay, I've shown you we can make this aerosolized in the lab when we take the water back to the lab. Can we actually get it to go up into the, do we see it in the real atmospheric, conditions? And so we actually went around and you can see the PM 2.5 conditions at the different locations, particularly by the spillway. This are wind direction and wind speed. And when we were just in a boat, essentially driving around the lake, getting these different samples. This is over where the people are using the aeration systems. This is by the spillway. And just for context, this is, in picograms per meter cubed. If you have a toxic species, that's a pretty decent concentration for the atmosphere. So this isn't, just a negligibly small amount. Here's a picture of the spillway where we saw a whole lot of that stuff getting generated. Of course, at certain times of year, the water isn't going over the spillway, but at other times it is. You can't really see it in here, but you often have a whole lot of fish kind of just like flopping around on the bottom right below the spillway. So it's an interesting environment. So our question was, all right, here you can see just a little wave breaking on the beach, a little video I took. It's kind of fun. And we wanted to see, how much of the toxin were we seeing? So this first measurement we made back here, this one was made with only pulling about 3 liters a minute through a filter. So it actually isn't pulling a bunch of air, which is why we had no detection on these two right here, just for context. Here we're using a much heftier pump. We're pulling about 100 liters a minute so that we can get more air and get better quantification. And you can see our concentrations here for the microcystins at a couple of different locations on the porch of our townhouses, as well as on that beach. And you can see our, a little video there. We looked at this as a function of size. And so at the smallest, in the morning, right before we had a storm here, this is a thunderstorm rolled through, a little less severe than yesterday's, but of the same sort. And you can see that we had a lot of aerosol here, both before and after. Afterwards, it was actually a little bigger, which was kind of interesting. But then this is right on the beach, right next to the water where this is, the sampler was right next to this different sampler that you're seeing. And we see a lot of aerosol getting generated that's, sort of in this, what we call accumulation mode that stays up, for days to weeks in the air. So this LR and RR, is we're seeing a decent amount of that getting up into the air. So we also teamed up in this work to see, well, not just do we have toxin, but do we have any other material coming from the HAB? And so we teamed up with Lindsay Marr and her student Jin Pan here. And they collected and did rRNA, got data for that. And you can see this is the gene copies for different samples. And so they also had detectable levels of 16S rRNA, which is associated with the bacteria that are in the lake. I'm not a microbiologist, so I'm not going to go any further into that data. You'll just have to take my word for it. But yeah, we were pretty excited to see that as well. Okay, so I hope I've convinced you that we can see aerosol. I hope that we see aerosol that contains toxin. And one of the things we wanted to do next is say, okay, this is great to pull things through a filter and go extract it and look at a bulk sample, but we want to look particle by particle. Go back to that microscopy stuff I was talking about earlier and see if we can see it. And so we collected some samples onto microscopy substrates. We actually often just put it onto a silicon wafer that we break into little bits and put inside of our collector. And we take it back to the lab, and I'm going to tell you about these two methods that we like to do to look at individual particles that are really small. And so one of these is called atomic force microscopy coupled with photothermal infrared spectroscopy, which is a mouthful, but the basic gist of it is, if I have a particle right here, and I can bring across a little tip and just tap my way across it, I can figure out how tall it is, I can figure out what its phase is. That's not super rare. What's unique about what we're doing is that we'll then bring in a different laser right here, and we can sweep across the whole mid-infrared where you would normally collect an IR spectrum like you're in Orgo lab or something like that. And if the particle happens to have a mode that absorbs that, it will expand. Because you think about an IR camera or any sort of infrared, that's heat, right? So as it heats up, it expands. When it expands, the tip goes up. And we can actually back an IR spectrum out of that. And so here, just for example, this is normal IR, FTIR here in gold versus red is an AFM-IR, where you can see ammonium and sulfate lining up right where they're supposed to be. So the benefit of this is that unlike a normal IR microscope, where you can't really get smaller than say 10 or so micrometers, Easily, honestly, below 30 is kind of tough. Here, we can get down to 30 nanometers. So we can see really, really small things and get an IR spectrum. But sometimes slime is gooey and it can gunk up your tips. So that's kind of unfortunate. So what we do in that case is that we bring down a green laser. So we take that tip that was tapping across and we get rid of that. And now we just have a green laser coming in here and it's scattering off of there. And if the thing does the absorption, it will scatter more light. And then we can actually back out an IR spectrum from that as well. And so this gets us about 1 micron or so spatial resolution. So still an improvement, but not 30 nanometers. And so here, I won't bore you with the physics equation, but things get bigger. That's the basic gist here. But what we can do is we can see really nicely, say ammonium sulfate here is our standard. We have both the ammonium peak and the sulfate peak. The other nice part about this is that we also still are going to be able to do normal Raman microscopy off this as well. And those inelastically Stokes scattered photons will end up giving us a Raman signature of that exact same particle simultaneously. So we can do IR and Raman at the exact same time, which is pretty powerful. And so let's think about how we can use this to look at stuff that's coming out of our algal groups. And so here's our microcystin LR structure. And if you've done IR of biological molecules, you might be used to looking at, say, the amide 1. So that's where we have carbon and oxygen. We have our carbonyl mode. If any of you have ever taken this chemistry classes, you dance with the different vibrations. So here we're looking at the oxygen going in and out at these yellow positions. We also have the amide 2, which is an NH bend. So that's more of a motion kind of like this, as well as our CN vibrations for amide 3 that are a little less prominent. And so we compared this with your normal FTIR. So if you were to just take an output of water, let it dry it out, and go run IR on it, this is what you should see. One of the challenges you run into with IR often actually is that water will interfere in this region based on one of those terms in the physics equation. Water is not super active for photothermal infrared, so it actually lets us get a much cleaner amide 1, which is really useful. And so here you can see our optical one with the green laser, as well as our one with the AFM tip. And in all of these, we get a really nice amide 1, which is kind of what we're looking for. So let's look at something that we all find interesting. Here we've got Planktothrix. And so Planktothrix, this is a sample from Grand Lake St. Mary's. Our collaborator, Judy Westrick, gave us this this data is pretty recent, actually. It's about 2 weeks old. So hot off the press, you could say. And so here you can see that nice filament of the Planktothrix with an SEM image that we went up and collected. And when we go and get the IR spectrum of this, we can actually end up seeing that really nice amide 1 right where it's supposed to be, as well as some other modes. I'm not going to go into all the fun spectral labeling. But the other thing we can do with this is we can do mapping. And so we actually can map here one of these Planktothrix. This is looking at this region. And we get a really nice map for that 1650 that you can see there for the amide 1, as well as the amide 2. Not quite as intense, and there's a background that's perfectly purple and flat. So it looks really nice in that sense. Now we can compare that to what if we have sort of a mixed biological organic particle. We can still see that clear amide 1, as well as other peaks that are associated with the organic material. So we can kind of tell you how much biological material might be in that individual particle that's floating around. And so one of the things we wanted to look at was if we take some of this water that we now have a way to characterize, and we look at it before we have lysed it and after, how much stuff is going to get up into the aerosol phase. And so here, this is our raw water, so straight from the lake, essentially, not the other connotation of raw. And we take that water, put it into our little aerosol generator, and we can make some microcystin aerosol. But if we lyse it, so we just did 3 freeze-thaw cycles on this, we end up generating a whole lot more aerosol, and we can see all the different congeners we were characterizing for that. But we can also then, for the individual particles, tell if there's more biological material in all of them. And so here you can see that for both the raw and the lysed water, the dark green here is biological. You'll see that that increases as we go from below a micron, submicron size, to supermicron, submicron, super. In both cases, we have more biological in the bigger particles, and we have more of this stuff when it's lysed. And so this actually compares really nicely to some of that initial work that I told you about with the single particle mass spectrometer. So totally different measurement, but it correlates nicely, more biological in the, we call the bigger sizes here 1 to 2 microns versus at the smaller sizes at less than a micron. So the trends track pretty well. We're very excited about that. Okay, so I think to tie that all together, pretty much we've shown you that bubbles matter. Bubble size matters. We make the aerosols. We see them in the field. We see them in the air, getting both transported and modified. And we can see toxins getting up into the air of different sorts and see them both at a single particle level as well as with nice concentrations from mass spectrometry analysis. So with that, I want to make sure to acknowledge all the great folks who have done the work here, particularly the early work. Nate May did a lot of this work. Our collaborator, Carrie Pratt here. Alexa is the undergrad with the camera. I didn't have time to talk about it, but we've actually published 3 papers with David Schmalley last year looking at autonomous sampling with drones over that same lake. So both air sampling, water sampling, and wind data. That's Judy as well right next to him. John and Nicole had most of the data from my lab that you saw. And this is us over on Prairie Creek sampling during one of these blooms in 2019. So with that, thank you all for your attention, and I'd be happy to take any questions.
Okay, thank you, Andy. Is this microphone working okay? Sounds like? Okay, great. So we do have time for questions, and we welcome questions both within the room as well as online. So I'm going to manage the in-the-room questions here, and I think Mary will let us know if any come in online. First question is from Reagan Herrera.
Yeah, of course I go first. So one of the questions that I actually get asked a lot now is, should our people be wearing masks or protection out in the field knowing this, especially some of the data that you showed? And is like a regular mask that we use, it looks like, because they're larger particles, enough to potentially protect us when we're out there, do you think? Based on your research?
Yeah, that's a great question. If it was, I guess I would answer in terms of what I would do personally. So and I'm a cautious person clearly. So if it were a windy day and there was a decent amount of wave action, I would wear one personally. Pretty much any sort of KF94 or KN95, any of those, like, the kind of thing I'm wearing would totally work. Those sizes are pretty easy to filter out by a mask once you're above a micron. So yeah, if you wanted to take that layer of protection, that would definitely work.
Great. And then I have a follow-up question, if I can go ahead. So some of the nutrient stuff that you were showing, how much do you think that potentially could be contributing to nutrients, specifically nitrogen, to the bloom as it's coming in?
Oh, you're talking about nitrate deposition?
Yeah.
No one has looked at that at all. It definitely could play a role, particularly I think kind of western Michigan, downwind of Chicago is an area where you have a lot of NOx converted eventually to nitric acid. And then possibly, yeah, sort of the south coast of Lake Erie would be another place I would think atmospherically that a lot of that would end up, sort of the impact of Detroit and Toledo and winter and stuff like that.
I think we should remember that for EcoHab.
Sounds good to me.
Yep.
Yes.
I'll give up my mic now.
Okay, thank you, Reagan. Any other questions in the room here? I had a quick question. It sounds like it's possible to get whole bacterial cells up into the air. Is that correct?
A lot of what we're seeing is not that, but yes, it is possible. Some of the stuff out at Scripps, actually, they've done some nice work showing intact cells going up. I would say as a fraction of the material that's getting up, it's probably pretty small, but it definitely can happen. And if it does, it would be, I think, really interesting to think about.
Yeah.
The other thing I didn't mention that probably is pretty important is that if you inhale these, you tend to be a lot more, the toxicity is estimated somewhere 100 to 1,000 times more toxic, the impact if you inhale versus ingest. Just because, our bodies are a little bit used to taking in stuff into the gastrointestinal tract. Lungs are not so used to getting these kinds of things into it. So from that context, it'd be really interesting. I don't know that anyone's looked at intact cell toxicological assays on this, or maybe people have. I'm not aware of it though.
Yeah, I think there's also interesting implications for dispersal of bacteria to inland lakes and the ecology of that. But my question was about the potential for those cells to nucleate snow in the winter. I know bacterial cells are great nucleators of snow. So could that play a role, especially in lake effect snow?
Yeah, we actually proposed that to DOE one time. We weren't able to get it funded, but yeah, no, Allison Steiner and I have talked about this, as well as we have some collaborators out of Colorado State who do ice nucleation as their main focus. One of the folks actually who I had Paul DeMont, personal communication. Paul's probably one of the leading experts in ice nucleation. And those were wintertime flights over the Great Lakes. And he saw the lakes by aerosol up in the clouds. So I would say there is anecdotal evidence that it probably matters, but it needs to be explored further.
Great. All right, question from Abby.
I'm not going to talk about snow. Coming from a minimal chemistry background, but I am an atmospheric scientist, when you talk about aging of these chemicals when they're aerosolized or lofted into the air, how much impact do the atmospheric conditions play a role? Does temperature affect how these age or move about, or does solar radiation play a role? Or I guess with that question in mind, I'm thinking about in terms of how our climate is changing, do we expect to see these concentrations change as well?
Yes. So as somebody who comes from an atmospheric background, I appreciate the chance to talk about temperature, relative humidity, pH, all these other things. The temperature of the lake's getting warmer. I think, as you have a more abundant bloom, you're going to get more stuff up. Wind speed and wind direction play a huge role. If you're downwind of the lake versus upwind, I would expect you're breathing in a whole lot more of this. Also, wind speeds are predicted to increase over the Great Lakes, at least some of the papers I've read over the next, by the end of the century. So that's going to kick more stuff up. So wind speed, wind direction would play a pretty big role. Solar, I don't expect to have a huge impact while it's up in the air. Their molecules are relatively robust. You're more likely to get chewed up by oxidants in the aerosol actually reacting with it, like OH radical, and you can have nitrate radical doing things. It's something I think there's been one study, I think, where somebody tried to look a little bit at LR aging in aerosol with OH radical. And I think it was relatively robust, but it did eventually break down. Were there other variables you threw out there? I think I hit the main ones. So yeah, I would expect more stuff getting up. And I think in terms of how it evolves in the atmosphere over time, I think that's something that we need to do some more work on that I don't have a good answer to.
Any other questions? If not, then let's thank Andy one more time. Thanks, Andy.
Thanks. That was fun.
Yeah.